So good morning, everyone. Good afternoon for those in the Netherlands. So it's my great pleasure to introduce today's seminar speaker, Dr. Eric Poelman. Eric is a scientist I greatly admire and whose work I follow regularly because of our common research interest. I had the opportunity to visit Eric's lab during my sabbatical 6 years ago, and it was one of the highlights of my sabbatical stay. Eric is an assistant associate, or I think it's associate professor at the Laboratory of Entomology at Wageningen University in the Netherlands. He obtained his PhD in 2008 from the Netherlands Institute of Ecology at NEO under the supervision of Dr. Louis Vet. His dissertation was on the metabolomics of indirect defense in cultivated Brassica oleracea varieties and its effect on insect biodiversity. At Wageningen University, his research team studies the dynamic interplay between plant defense plasticity and insect community organization, parasitoid host location in complex environments, and plant-mediated interaction networks of herbivores, their parasitoids, and hyperparasitoids. His projects combine ecological, modeling, genomic, and chemical approaches to answer complex community-level questions on the roles of plant defenses. As an interesting note, Eric was trained as a vertebrate ecologist and worked on the taxonomy and reproductive biology of poison frogs. So today he will talk about his work on multiple herbivory and plant defenses. So Eric, the floor is yours.
Thank you, Cesar. Very nice introduction. We'll start sharing my screen. Let's see. I hope you can all see my screen. Yeah, indeed. I have a background in vertebrate ecology, so you can see some of the vertebrates that I've been studying in the bottom of my slides. These very beautiful colored frogs that I still like to study if I have the chance. I keep some of them at home as well, so still interest in this. But now my work really focuses on plant-insect interactions. And I try to cover a bit the flavor of what we are doing in my team. And I have titled that as Plant Defense to Attack by Multiple Herbivores. I will also show you a bit of work on parasitoid wasps and pollinators. And well, I think what you will recognize in this talk is also that I got inspired a lot by the time when Cesar was doing his PhD. Many of the flavors of the work that he has been doing at that time with Jennifer Thaler, with Anurag Agrawal. So they have been a great inspiration for the type of work that I'm doing today. And if I look at plant-insect interactions, then I see plant-insect interactions as really these community processes that are taking place on these individual plants. Individual plants are interacting with a lot of different species. And one of the things that I feel is really important to add to a picture of the interactions is that these interactions are also structured in terms of time and space. And if you, I see that the presentation is not moving forward. That's not good. Maybe I'll stop sharing and start it again. Yes, hopefully this works better. So if I look at these systems, then these individual plants, they are growing in another vegetation of plants around them. So what is important is that these individual plants, their environment where they stand and where they grow is constantly changing around them. They have to compete for light, for nutrients. And at the same time, what you will see is that over that growth season, for example, here, black mustard plant that's growing along the River Rhine, you will see that it gets colonized by a whole suite of different herbivore species. And what I find very important is that not all herbivores come at the same time. They come one after the other, and they start to attack the plant in a certain sequence. And that's why I would really take the viewpoint of that these interactions are very dynamic systems. So they are dynamic systems of plants growing, but also these insect communities around them. They are dynamically changing. And why is that so important to realize? Well, if we are to study plant defenses, then we know that plants defend themselves constitutively. So they have a certain level of secondary chemicals that make them resistant against insects, or they may have physical characteristics like trichomes that make them resistant. But what
Eric,
we also know...
Eric, sorry, but we're still seeing the first slide.
That's
I was
funny.
wondering.
I am moving forward. Let's see how that works then. I'll see if I can.
Can you make the slide full screen?
Yes. Can you now see the full screen slide?
No.
No.
Oh, that's funny. I don't know what's happening.
Well, now it's moving forward, but we're still seeing. We're not seeing them in the full screen.
Okay. So if I now set it to full screen, can you see a picture with a Pyrrhus rape butterfly and a Nebraska Niger plant? Or do you see something else?
You see your second slide. And we're still seeing as though you're working on your talk. Okay, now we're on slide 3.
Yeah, I think something goes wrong with the I stopped sharing. I'm sorry for this. See if I can share again. So do you see my...
That looks better.
Yep, there you go.
And now it also moves to the next one?
Yes, that's
Yes.
slide 2.
Perfect.
Great. Well, I'll skip the little movie that I made, which is the plant that was growing in this environment. And you see these insect species that are arriving over time. And we basically, what I conclude here is that what is very important is that there's dynamics in these insect communities, and that there's dynamics in the environment where this plant is growing. And why was that so important? Well, it's really important because what we know is that plants defend themselves constitutively, but also that plants respond to herbivores. And the reason why plants respond to herbivores is that paying constantly the cost of constitutive defenses comes with the cost that you cannot grow very quickly. So this makes that plants are optimizing their energy budget and only defend themselves more heavily when actual herbivory takes place. And that's what we call induced resistance or induced defenses, that plants are responding to the herbivore. And these responses could include, for example, an increase in concentration of defense chemistry, like glucosinolates for brassica plants. But it could also be that trichomes are in a larger density. And what we see is that most of the time when the herbivore is feeding on an induced plant, the plant is responding specifically to the particular herbivore species that is feeding on this plant and tailors its defense to that particular species that attacks the plant And we see that it decreases the performance of the herbivore that's feeding on that plant. But what is very important is also that if the plant changes its phenotype towards one herbivore, it will also be viewed by everyone else in the community as a different plant phenotype. So the interaction with all sorts of other organisms will change. And what we see is that, for example, a response to one herbivore in terms of changing your defense chemistry or changing your trichome density affects the resistance that you have as a plant to a second herbivore. And what you should then conclude is that herbivores are interacting with each other via the plant because herbivore A changes the plant phenotype and thereby affects herbivore B. And that's what we call plant-mediated species interactions. And we realize nowadays that these interactions become very important in how insect communities are structured and how plants should take these factors along when they are defending themselves to herbivores. And some of the work that really inspired me is the work that Anurag Agrawal did on milkweed plants. And basically what they showed was that if you have a plant that is challenged early in the season by a weevil or by a monarch butterfly or by a leaf beetle, then everything that colonizes this plant after that early season herbivory is going to change. So what you see in this figure are two very important things. First of all, different herbivore species that were challenging the plant early in the season had a different effect on the richness of species that was going to colonize these induced plants. And the second thing is that these patterns may not be similar or exactly the same each year. So in 2001 and 2002, when they did their studies, they saw that the responses of other species were a little bit different. So what is important is that we have this context dependency of these interactions, but we also see that basically the community that colonizes a plant is affected by the first herbivore that arrives on the plant because that first herbivore is changing the quality of that plant. And if we start extrapolating this importance of these interactions between herbivores, we could argue that they might also be very important for the evolution of plant defenses. And there, one of the classical papers that I would like to highlight is work by Scott McCart. He actually showed that if you have a leaf herbivore feeding on the plant, it would affect the likelihood that this plant is going to be attacked by a seed-feeding herbivore or flower-feeding herbivore. And what you see is that if the plant was first attacked by this leaf-feeding herbivore, you get fewer individuals of the second species attacking the reproductive tissues. And if you start to think about this more into an evolutionary perspective, you might start to argue that actually herbivore 1 that's feeding on the leaves has a very important effect on the reproductive success of that plant. So it could mean a lot for this plant, how it responds to that first herbivore, what eventually its reproductive success would be, not necessarily by that first herbivore, but because of all the effects that later on happen on this individual plant. And my work, the work in the team that I lead has tried to explore a little bit more of these type of questions. And nowadays, we're still doing these type of experiments, trying to unravel interactions between plants and insects all the way in these dynamic patterns towards plant fitness. Here you see an example of one of these studies that I did, is that we were looking at a perennial plant, a brassica, a wild brassica accession that we used, a Brassica oleracea, and we induced these plants in year 1 when they were still vegetative. These plants were induced by either leaf chewing or sap feeding herbivores, and we left plants unchallenged. Then in the second year, these plants would start to flower after the winter, and then would be their first reproductive year. What we monitored was in both of the years, the herbivore communities that arrive on these plants. We measured a lot of plant traits to identify how the plant was growing, And in the second year, we would measure seed production of the plant. And what we wanted to identify is whether effects that happen in early season in year one, whether they translate all the way to next year when the plant would be flowering. And what we found was that indeed herbivores that were colonizing the plant in year one affected plant parameters in year one. So they basically predicted the growth and the number of leaves that this plant could make. And that translated to how well the plant would grow after the winter in terms of the number of leaves and the height of the plant, but also the number of flowers and seeds that the plant could produce. So what we could identify is that these plant-mediated species interactions also can happen across different years, across different seasons, even when there's a winter in between. So what we identify with that is that these patterns of interactions, dynamic interactions, are really important for the plant, even if you are a perennial. What we want to unravel more is then what sort of species are the most predictive of the reproductive success of the plant. You can imagine that all these indirect interactions take place on a plant. There are sap-feeding herbivores, leaf-chewing herbivores, below-ground feeding herbivores, all sorts of microorganisms interacting with that plant. There are natural enemies, there are pollinators, and all of them have an effect on the reproductive success of the plant. And we wanted to identify, can we predict which of these herbivores, or maybe the pollinators, or interactions between herbivores and pollinators, which ones are now the most important? And in one study, we especially focused on the interaction between herbivores and pollinators. And there we identified that herbivores, when they are feeding on the leaves or on the roots of a plant, change dramatically how the color of the flower is, how the odors of the flower are, how the shape of the flower is, how the nectar reward or the pollen can be affected. And because all of these effects of herbivores translate into quality aspects of the flowers, we see that individual pollinators are also going to interact differently with these flowers. So we see that plant-mediated interactions in a response to herbivores translate into how plants interact with their beneficial organisms, such as these pollinators. Well, currently, we had an interesting discussion already with Cesar's group here before, and one of the aspects that we are currently studying is also to identify better what it means for plants in terms of reproductive strategies, if they are accepting their own pollen or if they are outcrossing more under challenges like stress by herbivory. And of course, it's always then the question, when are these interactions taking place? When are they the most important? Is it really when the herbivores are present, when the plant is making flowers, and then it will affect the pollinators? Or can also herbivores that are feeding on these vegetative plants that have not yet made flowers affect how well this plant is interacting with the pollinators? Well, interesting work that Quint Russman did in my team was to identify if you are placing herbivores at different moments on a Brassica nigra plant during its vegetative early life stage, or during the stage when it was going to make its first flowers, or when it was already fully flowering, how strongly will it then affect the interaction with pollinators and with seed set? And what he identified is that especially the events that happen very early in life of the plant translate the strongest to effects on the pollinators and on seed set later in life of the plant. So very early events were very important for what we see in reproductive success later. So you can imagine that plants have to take along all these type of processes in reaching maximal seed production. Well, if I then look at how we are currently viewing how plants deal with insect damage, then we know quite detailed how plants respond to insect feeding. We know, for example, that there are markedly different strategies of plants to respond to aphids or to caterpillars. For aphids, we know, for example, that the phytohormone salicylic acid is really important, and the whole signal transduction cascade that is regulated through salicylic acid is an important aspect in how plants defend themselves or regulate defenses against this aphid. For caterpillars, we know that it's more important that the jasmonic acid route, and that determines how well the plant defends itself against this particular group of leaf-chewing herbivores. Well, in a lot of work, we also have identified that these two pathways of the salicylic acid and the jasmonic acid pathway cross-communicate with each other. So things that happen in the salicylic acid route will affect what the plant can still do in its jasmonic acid route. And quite often this was seen as an idea that plants can actually fine-tune their responses if they have to deal with multiple different type of stressors, so that they can still manipulate or still maneuver between all these different attackers. But what we also identify is that salicylic acid negatively affects jasmonic acid. And especially in studies on Arabidopsis, it has been identified that these processes are very strong, and that it could happen that if aphids feed from the plant, it actually impairs the jasmonic acid responses to caterpillars, and caterpillars would grow better on plants that were first attacked by aphids than on control plants. And that aspect I find really striking, because it feels like one plant is defending itself against a particular species of herbivore, and it impairs the possibilities that it has to deal with other species. Suppose you are a plant that's always attacked first by aphids and then by caterpillars. Would it really be adaptive to not be able to defend very well against this caterpillar? So I started thinking a little bit more like, okay, what if these plants are now attacked by a lot of different insect species? And surprisingly, there's very few studies that have focused on plant response to more than 2 herbivore species. So we did a first study to start and challenge plants with either 1 species, 2 species, or 4 species of herbivores. And we started to play also with the identity of these herbivores, with in the back of our minds the idea of this salicylic acid cross communication with jasmonic acid. So we challenged plants with either only aphids, 1, 2, or 4 different aphid species, only leaf chewing species, 1 caterpillar, 2 caterpillars, or 4 different caterpillar species, or we did a mix of these two, that the plants were attacked by an aphid and a leaf chewer species, or by 2 aphids and 2 leaf chewer species. And then we induced these plants with these combinations of insect herbivores, and then we fed the plant to yet a new herbivore, the diamondback moth, to monitor basically how the plant has changed its possibility for resistance against a late arriving herbivore when it had already dealt with either 1, 2, or 4 different herbivore species that were more similar in their traits, all being from the same feeding guild, or being more different, being from different feeding guilds. And if you look at the graph at the lower left, you see basically that for caterpillars, for leaf chewers, the blue the blue dots, you see that when the plant is attacked by 1 leaf chewer or 2 leaf chewers or 4 leaf chewers, it didn't matter too much how well it was resistant against yet another new leaf chewing species, the diamondback moth larvae. It would be able to still defend itself very well, and even has higher resistance against that leaf chewing species than a control plant. If we challenge the plant with 1, 2, or 4 aphid species, we see that first of all, when aphid species were feeding on the plant was indeed less well resistant against the subsequent attack by this Plutella caterpillar. But especially when we put 4 different species of aphids, this effect was stronger. So if we had a diversity of 4 different aphid species, the plant would be more, would have more difficulties with defending itself against a next leaf chewing species. And then what we see in the green bars is basically that when the plant was challenged by a combination of aphids and caterpillars, it would have the same resistance as what it would have when it was unchallenged before. It would be able to still respond very well to that diamondback moth caterpillar. So basically what you see is when you challenge the plant at the same time with two feeding guilds, one of the guilds is not overriding the effect of the other, but basically it's integrated into a response that still allows the plant to deal with a leaf chewing species. So what I conclude from this work is that yes, indeed, it matters a lot how many different species attack the plant and how similar or dissimilar these species are in their characteristics. But you can argue, of course, that now I've been challenging plants with everything at the same time. We're currently doing a lot of studies to identify what if you are challenging a plant with a sequence of 4 different herbivore species and you change the order of arrival. We very recently published a paper with the first results of that work that I'm not showing here today. But what I take from that work is that it matters who was the very recent herbivore that attacked you. That determines strongly what your capabilities are as plant to still deal with the next herbivore, but also events that happened much longer ago, like the first herbivore that arrived on the plant in the sequence, does determine what you can do against even a 5th herbivore that's arriving on the plant. So the sequence of all of these herbivores is really important in what the plant can still do as a defense. And I think the challenges that we have now is to identify what plants really can do in terms of plasticity and what sort of plastic strategies plants eventually have. Well, we challenged also the idea of that a plant responds to aphid feeding and whether that impairs its capabilities of dealing with caterpillars. And what I already concluded was that suppose you are a plant that is always attacked by aphids first and then by caterpillars, it's hard to argue that plants are going to be impaired in their response to caterpillars. That seems like not a very adaptive strategy. So what we did was to challenge basically that idea, and we used the Brassica nigra plant and 10 different herbivore species and did a matrix of all pairwise interactions of these species on that Brassica nigra plant to see how that plant regulates its responses to 2 different attackers. And what you see is that we here did a matrix in different colors. At the bottom, you see the inducer. It's the first herbivore that we put on the plant. And the color gives the performance of the second herbivore that we call receiver, that's on the y-axis. And what you see is basically, if we argue from the idea of that plants really respond to the first herbivore, and that affects very strongly what it can do against the second herbivore, you would argue that the colors in the columns going up from your inducer would actually have very similar colors. And if you look for every herbivore and going up to check all its colors, you would see that there's quite a diversity in colors, so that there's not a very clear pattern that is determined by the inducer itself. And that could be because we challenged the plant with different receivers as well. So we had leaf chewing species and we had sap sucking species. And you might think that if we have a leaf chewer, we would actually see similar colors for the leaf chewers, but different colors for the aphid species, the sap sucking species, but also that didn't happen. So there was not a clear pattern based on the identity of the first herbivore. If you then focus on actually the x-axis, you would see that the focus from the receiver herbivore, the second herbivore that's feeding on the plant, the colors start to become more similar. So we see actually that the second herbivore was a better predictor in how well it would perform than actually the inducer that was on the plant. And also we would identify that not necessarily if aphids were feeding first on the plant, caterpillars would perform better on these plants. Sometimes we would find that they would perform worse. Sometimes we find that they would be neutral in their effect. So this challenges, I think, the idea of this salicylic acid effect on jasmonic acid as a predictor for the outcome of interactions, ecological interactions between species. We did measure also genes that were induced by these different feeding herbivores, and we indeed identified that based on gene expression, we could actually match literature in terms of how these different herbivores would induce salicylic acid related gene expression or jasmonic acid related gene expression. But what we then eventually conclude is that the gene expression is not a good predictor for the ecological outcome. So we're not challenging the idea of physiological interactions between SA and JA, but that physiological interaction may not be the best predictor for the actual performance of the herbivores. And what we thought was then, okay, but what is then the predictor of why plants are better capable of being resistant to the second herbivore when they had responded to the first one? And then we went back to field data. We collected insect arrival data on 3 years of a lot of individual Brassica nigra plants, and we scored the full every week how many herbivores were on that plant from which species. And from that data, we identified these pairwise interactions that we tested in that matrix. How frequent do they actually occur in nature? And then we identified that there is a correlation between the resistance of the plant to that attack by these two different species and the frequency of how often these interactions take place. The more commonly these interactions were taking place, the better the plant was adapted to this particular interaction. So it could get higher resistance against the second herbivore when it was an interaction that it would encounter more often during its lifetime. So there we see that plants apparently adapt indeed to the sequence of herbivores that is interacting with it. Well, based on these findings, we started arguing what this might mean for our research field in terms of plant interaction with multiple herbivores. And I think what we identify is for plant physiology that you should realize when you measure a response to one herbivore that's feeding on the plant, it's actually not the response only to that current herbivore, but you're measuring the response of that current herbivore in the context of how that response evolved for that entire community of species that the plant will be interacting with. So you could see a signature of what's going to happen in the future with the plant already in the response to the first herbivore. It could also be that on an ecological level, you might start to see that insect communities are going to be partitioned over individual plants. For example, one plant species as a whole could interact with 12 different herbivore species, but not all of these 12 herbivore species will arrive on every individual plant. And what could happen if plants respond to one herbivore, it might mean that you will get a subset of other herbivores that either avoid these induced plants or are attracted to these plants. So if you get herbivore A, it might mean that you also get B, C, and D, but maybe not E and F. And then eventually on evolutionary level, it might mean that plants might have interactions that they take along in evolution. So if a leaf-feeding herbivore always predicts how frequent you are going to interact with a seed-feeding herbivore, we might get linkage between leaf traits and seed traits or flower traits in terms of evolution. So I think if we are placing back the interactions that we study into community perspective, we should start to identify how these data that we have or the focus that we have had on pairwise interactions now translate into new concepts for community perspective. Well, I've been talking a lot about plant interactions with herbivores, but of course that happens again into a context of natural enemies, and these natural enemies also have their own enemies. For example, parasitoids have hyperparasitoid enemies. But what does complexity now mean actually for parasitism? Well, in a lot of studies, we've seen that if plants are being attacked by non-host herbivores, herbivores that cannot be parasitized by this particular parasitoid species, we actually see that that affects the efficiency of parasitizing the host herbivore. So non-host herbivores may affect the interaction, the trophic interaction between a host and a parasitoid. And we did that in small tent experiments to show that if you have a simple situation with only the host or control plants, undamaged plants, we measure parasitism rates. Then we compare these parasitism rates to plants that were standing next to non-host damaged plants, or we were mixing non-hosts entirely over the environment. And there we see that in both situations where you have non-hosts in the system, parasitism rates drop. So it means that non-host herbivores also affect how parasitoids actually manage to find their host herbivore. And that is also dependent on the traits of the non-host herbivore. If the traits are, for example, a leaf chewer versus a phloem feeder species, we see that parasitoids that are associated with leaf chewer herbivores are affected more strongly by non-hosts that are also leaf chewers than that they are affected by non-hosts that are phloem feeders. And that could, for example, simply be that leaf chewing species induce very similar volatile blends that the parasitoid has difficulties with to discriminate. So here you see wind tunnel tests where we tested a parasitoid choosing between a plant damaged by a non-host or one that has a host and a non-host. And it could most of the time smell out the host species, but what we would see is that the non-host would also attract some of these parasitoids to plants where there were no hosts. So that determines also that the efficiency of parasitism may drop. So also here in these type of interactions, we have to place them back into community context to really understand what's happening under the complexity of natural interactions. Well, then, of course, we can have the focus on the hyperparasitoids, because also they are foraging in these environments. And we asked the question also how these hyperparasitoids are now capable of finding these parasitoids. And we realized that also here plant-mediated interactions are really important. We were questioning how these hyperparasitoids find their hosts, and hyperparasitoids, they lay their eggs inside the larvae or pupae of other parasitoids. And we have true hyperparasitoids that do that by basically penetrating through the herbivore, reaching the larvae of the parasitoid and laying eggs inside these larvae, or hyperparasitoids that we can call pseudo-hyperparasitoids that actually parasitize the cocoons or the pupa of these parasitoids when they have already left the host. But you can imagine that in both of these situations, the larvae are difficult to find. They are not interacting with the plant. So it's very hard for these parasitoids to actually use a very clear cue that predicts where these larvae are. But what we also noticed is that when parasitoid larvae are growing inside herbivores, the color of the spit of the caterpillar is going to change dramatically. So without measuring anything in that spit, we could already see that the color green predicts that parasitoids are doing a whole lot to the herbivore in terms of physiology. Then if you apply that spit to plant damage, you would also see that the plant is responding differently to these different spits of a healthy caterpillar or a parasitized caterpillar. And what you see here, just for one gene, we did that for many different genes, you can identify even the different species of parasitoids that are growing inside the herbivore that actually determine the signature on the plant in terms of its response. So we found consistent patterns of different parasitoid species growing inside a herbivore. And for two different herbivores, we find that each parasitoid does more or less the same thing to plant responses. So parasitoids change how the herbivore is interacting with the plant. And we identified that actually that is an important element for hyperparasitoids to respond. The changes in oral secretion by the parasitoid in the caterpillar is eliciting a different response in the plant is going to make a different odor blend, and that makes that the hyperparasitoid can smell whether on a plant there are now parasitized caterpillars or there are healthy caterpillars. And well, we found proof of that by basically asking the female hyperparasitoid in a choice test for the odors of the induced plant, do you like the ones where there are the parasitized herbivores, or do you like the plant damaged by a healthy herbivore? And then most of the time you would see this hyperparasitoid choose for the plant with parasitized herbivores. Then Antonio Cusumano came to my group and he actually said, well, this is probably not the whole story. It's nice that the parasitoids are changing something in the herbivore, but there's much more going on, because parasitoids are not just injecting eggs into the host, they are injecting venom, and some species are even injecting poly-DNA viruses that live in symbiosis with these wasps. And what he could do was to take out eggs, venom, and this virus separately and inject them with a microinjection technique into the caterpillar host to basically partition the whole event of parasitism. And what you can see here in this work, what we did was to compare caterpillar feeding on plants, so volatiles induced by caterpillars that were injected with a PBS fluid, a mock fluid, just to mimic the injection technique, with a PBS fluid, a caterpillar that was injected with a PBS fluid that either was containing virus, venom, eggs, or a combination of those. And then the plant would be induced by these caterpillars, and we asked the hyperparasitoid whether it could discriminate between these two situations. The lowest comparison that I show you here in orange is the actual response that we already knew from an intact parasitized caterpillar. So the hyperparasitoid preferred that over the injected PBS fluid caterpillar that was not parasitized. And if we then mimicked the parasitism by ourselves injecting the full sweep of compounds that a parasitoid would inject, we could restore that phenotype very well. But if we were taking out certain elements, then we would see that we were losing some of the effects. And what we could identify, what is really important, is that we have the virus and the venom, but not so much the parasitoid eggs. So apparently it is not the parasitoid larvae that is changing the herbivore, but it's the poly-DNA virus that is actually an important mediator of all these effects. Then we did a challenging experiment where we surgically removed the salivary gland of caterpillars. And after that, the caterpillars would still be able to feed on the plant, but they would not have saliva. And there we could identify that if you remove the salivary gland out of a healthy caterpillar and out of a parasitized caterpillar, then you would lose the entire discrimination that the hyperparasitoid has for these two types of caterpillars. So indeed, we could confirm something in the salivary gland is changing because of parasitism. And because of parasitism, this is because of the poly-DNA virus that the parasitoid was putting in. But we're still trying to unravel this story all the way also to other sorts of microorganisms that are on and in this caterpillar to further identify how these interactions really take place. And we have identified that it's really complicated. When we see that it's actually microorganism mediated. Well, I would like to end a little bit with the flavor of work that we are also focusing on, and a challenge that we face nowadays. If you place all of these interactions back into our agricultural system, that we see we have to make more sustainable because of the negative effects of pesticide use, we're thinking of how we can actually optimize these agricultural systems where we do not use pesticides, but we use combinations of plants in intercropping systems, such as strip cropping, crop rotation schemes, or maybe even pixel cropping crops that were mixed for every square meter. And there we would like to see how we have to change that whole environment in maximizing the effect of, for example, biological pest control or optimizing plant performance by making use of ecological processes. Well, one of the things that I find very interesting there is that during my own PhD, I identified one cabbage cultivar that is more attractive to parasitoids than other cultivars. And we thought, if we can now mix these strips of your cash cultivar with a more attractive cultivar, can we then make sure that also the parasitism level on your cash cultivar goes up when you replace that in a replacement design of 1 out of 10 plants with an attractive cultivar. And what you see here in the comparison that we made was to compare a monoculture sole crop with an intercropping of these cabbages with wheat, intercropping with wheat and a nectar source, or intercropping with also an attractive cultivar. And there we see that if you add the attractive cultivar, we can increase a little bit the attractiveness of that entire strip to parasitoids. So we could increase biocontrol in these type of systems. Well, to conclude, the final thing that I think what is really an important challenge if we are taking all of these aspects that I've just mentioned into more an applied perspective where we are going to try and optimize sustainable agriculture, we need to start thinking about how we can maximize our cropping designs to benefit from ecological processes, place that in this multi-trophic interaction of herbivores, parasitoids, and hyperparasitoids, and to think about traits like attracting these different species, so being able to manipulate the behavior of the herbivore, the parasitoid, but also take into account the negative effects of the hyperparasitoid. Think about offering nectar, for example, in a clever strategy that is really only benefiting the parasitoid and not the other two compartments of the ecosystem. But also to place this into a landscape context, because in the landscape, we will find the parasitoids that we want to recruit. And we know that landscape elements are really important in actually being an important deliverable of whether there are parasitoids, yes or no. And we need to identify which landscape elements are really the best predictors of maximizing also biological control in our agricultural fields. So this is one of the challenges that I think we stand for now, and my team should also really make a major contribution to in the Netherlands. What I presented to you today is a lot of work from many people, a lot of collaborations, former group members, but also my current group. And I'm very happy with all of the work that they have been doing, and yeah, really proud of what they have achieved over the years. And with that, I would like to end my talk, and I'm happy to take any questions.
Do we have any questions? Cesar, did you raise your hand, or was that?
That was an applaud, but I can ask a question. So Eric, this is a great talk. Like there was a lot of things to think about. I think you know, we have discussed a lot of these ideas in the past, but one of the things you know, with multiple herbivory that that I was criticized personally when I was working on you know, on trying to manipulate multiple herbivores on plants was the density of the herbivores that you place on the plant. How important do you think is the density? Like, you know, having one aphid versus 100 aphids is different. So how do you know, work that?
I get your question. I think this is really difficult to say it. So once we are talking about two herbivores or more than two herbivores that arrive in a sequence, then we can play with all sorts of things. It's the density that can be important, the timing in between these herbivores, the order in which they arrive. And at some point, yeah, it's very hard to keep track of all of these variables that we have. I think density is definitely important. There is evidence that induced resistance by aphids to caterpillars can actually be there when you have a small number of aphids, but it might flip if you have a large number of aphids inducing the plant first. So it could be that a plant is actually also responding stronger if you have a higher density of a particular species. And that means that, it might prioritize maybe more a very abundant herbivore compared to a lesser abundant herbivore. So I think all of those elements we have to identify whether, this always is a dose response, or whether this is, has to do with prioritizing certain herbivores, or whether plants really respond differently to a particular feeding guild. And what I worry a little bit is that all of these factors might interact with each other, and that makes it very difficult, I think, to tackle what is now actually the most important factor.
Yeah, I agree. So there's a couple of questions. Liz, you raised
Yeah,
your
Liz
hand.
was first.
Hi. So I guess I kind of wanted to follow up based off that previous question, was making me wonder. So you had mentioned that having multiple, like a diverse amount of species of aphids will kind of exacerbate the plant's inability to resist future pests such as caterpillars. And I was kind of wondering if that was affected at all by like the relative densities of each pest. So like say you have 70% of one aphid and 10% of another and 20% of another, or like 60-40, or like would that change things at all?
Well, we haven't done that analysis. What we tried to do in that study was actually to keep density, our initial starting density, the same. So we started off with, if you have one species of aphids, we would, for example, put 4 individuals of that one species. If we had 4 different species of aphid, we would put 1 of every species, so that we would start off always with the same density. But of course, if you let that go for a while, then especially if you have a mix of 4 different species, you might get that one of the species is dominating more than others. And we haven't actually been able to unravel that, whether dominance of one particular aphid is actually is causing the effect. And I think it's not the dominance of one aphid species, because then we would have seen that the error bar, when we had one, so we did the experiment with one aphid, but we did that for all of the 4 species. And then you would expect, if one of the species is really important, that you get different effects for all of these 4 different aphid species. And then you would expect actually that you get quite a large error bar in your response. And that's not what we found for plants being attacked by one aphid species. So I think what you're raising is happening, and that maybe one of the aphid species might have a stronger effect. But what I also think that happens is that because you have 4 different aphid species that all have different effectors in their saliva, so you have something that is similar but still different, and that might strengthen each other in the response that this plant has to make. So it puts all its cards on the table for an aphid response, more likely when it has multiple species of aphids than when it's just one species. But yes, we should do the experiments really to play with density changes of these combinations as well, ratio changes. Yep.
Awesome. Thank you so much. Yeah, I'm a lot more curious about like how the different aphids sort of impact each other and how like one might strengthen another. So I'd love to hear more about that. Thank you. Your talk was awesome, by the way, very comprehensive.
Thanks.
Okay, I think Kasia had her hand up next.
Yeah, hi. So I had at first the saliva thing with the color differences that can identify parasitism or hyperparasitism is like one of the coolest things I've heard. But my question was about, so the hyperparasitoids are attracted to the volatiles that the plant is releasing. Has there been something that's been looked at into a way to like mask it, kind of in the idea of like mating disruption, where it can confuse those hyperparasitoids from actually being able to find the
Yeah, so if you think in terms of, so you can think in two directions. You can think of more evolutionary perspective. So parasitoids might be selected not to reveal themselves to hyperparasitoids, so they would benefit from, still being inconspicuous in the host, so that their host is not making that particular signature. And that's a little bit of evidence for that we have for solitary parasitoids that were not so easily recognized by this hyperparasitoid. So maybe the solitary parasitoid manages to not reveal itself through plant volatiles. And that could be because it manipulates the host differently. If you think more of an applied perspective, then I think one of the challenges, especially in greenhouses, that we see is that in aphid systems, for example, in pepper production in the Netherlands, we see that aphids are controlled a lot by biological control. We release a lot of parasitoids in pepper production, but that we get also hyperparasitoids coming into that system. There's a couple of approaches that we use there, or that we are developing basically to tackle these hyperparasitoids. And one is actually to identify what are the odors that these hyperparasitoids respond to, and try to make lures so that we can trap the hyperparasitoids. But of course, we want to trap only the hyperparasitoids and not the primary parasitoid. So it should be an odor source that is only attractive to the hyper and not to the primary parasitoid. And this is really challenging. And you could think of all sorts of banker plant approaches where you could try to lure the hyperparasitoid away from your crop, and basically see if you can remove the hypers from the system in that way, that you remove your banker plants now and then. But this is really one of the applied perspectives from this type of work that I can see that should be done.
Are hyperparasitoids more generalist, or are they specialists?
That's a hard one.
A loaded question there.
Yeah, I would say that most hyperparasitoids are more generalistic than primary parasitoids, but you do have some more specialized hypers. So in the aphid system, we see that a lot of hyperparasitoids are very generalistic. In the caterpillar system, we have some more specialized hyperparasitoids, but also a lot that are a bit more generalistic. Yeah.
Okay, great. Thank you so much.
Cesar, you're next.
A very quick question, Eric. I don't want to take all the time, so Jay can ask the question too. But I'm also very interested, fascinated by the saliva of parasitized caterpillars. So one of the things that Gary Felton finds with the microbes is that the microbes are activating supposedly the salicylic acid pathway that makes the caterpillars, the plants, more susceptible for the caterpillar to feed on them. Do you see any effects with the parasitized caterpillars? Like, do you see any effects on the microbes that affects how the plant is responding, like with the phytohormones?
Yeah, it's. I think this is a really difficult question. We had a lot of discussions, especially with Heiko Vogel in Jena, Max Planck Institute, and we were arguing that I think what Gary Felton especially shows is that this works for beetles, and these beetles, they have specific glands in which they actually have specialized microorganisms that help them suppress the plant defenses. But what we think with caterpillars is that there's not so many microorganisms, at least that's what we think, in the saliva. Maybe there are in the regurgitate, so in the foregut, that also might come in contact with the plant. But we know that these are really transient, and that there's a lot of variation there. So we're not so sure whether caterpillars are carrying really beneficial microorganisms. And in the salivary gland, it seems unlikely that they are there, because plant the caterpillar really tries to keep that as clean as possible. It doesn't want to have any pathogens coming in. So there's really a strong defense basically in not getting microorganisms in your saliva. But we haven't seen any obvious microorganism as what Gary Felton's group has shown. But what we do see, or what we at least have noticed that happens, is that also the virus particles of the poly-DNA virus that the parasitoid is injecting can enter the salivary gland, but then through the hemolymph and then enter the gland. So we're not so sure what happens there, if something directly from the PDV virus actually affects the plant, or so there's a lot to explore. That's for sure. Yeah.
Okay, Jake.
Hello, can you hear me?
Yes.
Hi, I thought your talk was really interesting, and I wanted to build off of Kasha's question on hyperparasitism. You said that there was a cultivar you worked with that was particularly attractive to parasitoids, and I was curious, was it also attractive to hyperparasitoids? And I wanted to know if you had any way to quantify this hyperparasitoid's effect on a biological control system.
Mm-hmm. So your first question, yes, we tested that, and we identified for 2 hyperparasitoid species that they actually had the same discrimination. So they also liked the cultivar that was more attractive to the primary parasitoid, was also more attractive to the hyper. So apparently they might be using very similar cues there. So for the hyperparasitism, how do you quantify the effect? Well, what you can measure quite well is the number of hyperparasitoids that emerge, or the fraction of hyperparasitoids that emerge from cocoons of the primary parasitoid versus, or for the aphid system, for example, from aphid mummies. And what we see is that hyperparasitoids in some systems can even up to 90 to almost 100% emerge from the cocoons or the aphid mummies. So hyperparasitism, especially in greenhouses, we've identified that it can actually be very high. And my own work in the Pyrrhus caterpillar system with the hyperparasitoid associated with Cotesia parasitoids is actually that the gregarious Cotesia parasitoids, each individual cocoon, I think in total, about 60% yields a hyperparasitoid instead of a primary parasitoid. So hyperparasitoids really can hammer the population of primary parasitoids.
Thank you very much. That's impressive, but scary.
Yeah.
Okay, so I think we are past 1 o'clock. I think we will adjourn seminar and move to the class part of our session today. So for all of you that came for the seminar, thank you for coming, and hopefully we'll see you again next week. And Eric, if you want to stop sharing your screen.
Yeah. Then I stop sharing.
Yep. Cesar, do we want to take a quick 5-minute break?
Yes, let's do that.
Okay.
Bye.
Let's start 10 after 12.
That works.